Welcome to This Explainer. Today, we're going to dive right into something absolutely mind-blowing. We're looking at how the grand, sweeping complexity of all life on Earth literally boils down to a microscopic chemical code. We're examining the hidden architecture of our very existence, and as you'll see, it all hinges on the absolute smallest of margins. Seriously, think about this for a second. What if the massive difference between a temporary throwaway message and a permanent genetic blueprint came down to just a single atom? I know, it sounds a lot like science fiction, doesn't it? But it's actually biological fact. A massive functional difference in every single one of your cells hinges on the tiniest chemical variation you could possibly imagine. Okay, let's jump into Section 1, Anatomy of a Nucleotide. So, what is a nucleotide? It's basically the fundamental building block of nucleic acids, like DNA and RNA. Think of them as the literal, microscopic Lego bricks of life. They consist of a 5-carbon sugar, a phosphate group, and a nitrogenous base. And much like a limited set of Lego pieces can build practically infinite structures, billions of these fundamental units polymerize together to build the complete instruction manual for a human being. Let's zoom in and look at these units up close. You'll notice there are 3 very distinct components. Right smack in the middle, we have a 5-carbon sugar ring. Attached to one side of that ring is the phosphate group, which you can see highlighted in orange, and over on the other side, in blue, is the nitrogenous base. Every single nucleotide in your body follows this exact, incredibly precise architectural plan. Now, how do these individual pieces actually snap together to form a chain? Well, the phosphate group of a single nucleotide connects to its 5' carbon. To link up to the next piece in the chain, the 3' carbon attaches to the phosphate of the next nucleotide, creating what's called a phosphodiester bond. This specific directional backbone dictates that our genetic code is exclusively read in one direction, from the 5' end down to the 3' end. Moving right along to Section 2, DNA versus RNA, the Great Divide. We all know about DNA's famous double helix, right? Well, when DNA forms that shape, it relies entirely on Watson-Crick base pairing. The nitrogenous bases project inward and lock together perfectly. Adenine always pairs with thymine using 2 hydrogen bonds, while cytosine pairs with guanine using 3. This amazing interlocking mechanism across anti-parallel strands is the entire foundation of our highly stable genetic code. RNA, on the other hand, plays by a slightly different set of rules. For starters, RNA uses a ribose sugar, which was actually discovered way back in 1891 by Fisher and Palade. DNA uses deoxyribose, which wasn't discovered until decades later in 1929 by Phoebus Levine. Structurally, DNA relies on that double-stranded helix we just talked about, while RNA is generally single-stranded, which allows it to fold up into all sorts of complex 3D shapes. There's also a base swap. RNA trades thymine for uracil. But you know what the real game changer is? It's hidden right there in the names of the sugars themselves. And that brings us to Section 3, the climax. The one-atom difference. 2'. That right there is the magic number. The entire massive biological divergence between DNA and RNA happens exactly at the 2' carbon position on that little 5-carbon sugar ring. Here's the big reveal. RNA has a hydroxyl group, which is an oxygen and a hydrogen together, sitting right at position 2' on the sugar ring. DNA, true to its deoxy name, lacks that oxygen entirely. It only has a lone hydrogen atom. Now, why does that matter? Because that single extra oxygen atom makes RNA susceptible to hydrolysis. It makes it a highly flexible, temporary messenger that's prone to breaking down quickly. DNA's lack of oxygen at that exact spot makes it incredibly chemically stable, which is absolutely perfect for permanent long-term data storage. The implications of this are just massive for structural biology. Check out this quote from a 2005 study published in the Journal of the American Chemical Society. For the longest time, the standard paradigm in biology was that this 2' hydroxyl group in RNA pointed away from the main structure because of steric hindrance. Basically, scientists thought it didn't fit. But researchers used advanced NMR techniques and found a real plot twist. This tiny OH group actually points inward. It creates this beautifully complex network of hydrogen bonds with surrounding water molecules, actively stabilizing the RNA structure in entirely unique ways. Just one single oxygen atom completely shifts the thermodynamic stability, even enabling RNA to act as a chemical catalyst. It's wild. All right, let's zoom out a bit for Section 4, Beyond the Genetic Code. You know, it's super easy to just think of nucleotides purely as letters in a genetic alphabet, but actually, scratch that, they are highly versatile multitaskers. They're really the unsung heroes of cellular metabolism and signaling. They don't just sit there holding blueprints, they physically drive the chemical reactions that keep our cells alive and functioning every single second. Take adenosine triphosphate, or ATP, for example. It's literally the energy currency of your cells. Structurally, if you look at it, it's fundamentally just an RNA nucleotide. It's got an adenine base and a ribose sugar with that same 2' hydroxyl group we've been talking about, but instead of just one phosphate group, it has a high-energy chain of 3. By simply slapping on a couple of extra phosphates, life turns a standard genetic building block into a super potent biological battery. Literally every time you move a muscle or even think a thought, you're burning through this modified nucleotide. And because we now understand these microscopic structural quirks, like the vulnerability of that 2' oxygen, we can actually synthesize modified nucleotides engineered to survive in our bloodstream. This has led to some incredible modern medical miracles. We're talking about using synthetic nucleotide derivatives to interrupt retrovirus replication in diseases like HIV and hepatitis. We can build antisense oligonucleotides to treat genetic diseases that were once thought to be completely untreatable. And of course, synthetic guide RNA serves as the precise targeting system for CRISPR-Cas9 genome editing. Which brings us to a really fascinating final thought. If changing just one microscopic Lego block, literally adding a single oxygen atom, can fundamentally redefine life, making the difference between a fleeting temporary message and a permanent blueprint for existence, well, how is our continued tinkering with these molecules going to redefine the entire future of medicine? We've really only just begun to unlock this microscopic code, and the possibilities are absolutely endless. Thanks for joining me for this explainer, and I'll catch you next time.